So greetings from Portland, Oregon. So I was going to talk a little today about just some of the exciting developments that are going on in the field. So as you may know, I'm an inherited retinal degeneration specialist. So I see exclusively patients with genetic eye diseases, most commonly retinitis pigmentosa, but also Usher syndrome, as well as a whole other variety of eye problems. But I've been quite involved with Usher syndrome and have a lot of interest in it. And I think there's a lot of exciting things going on. So I wanted to talk a little bit about what is on the horizon. So when we think about treatments for inherited retinal degenerations, they really fall into two different categories. So one would be treatments that are aimed at slowing down the degeneration or helping you keep what you have, and then therapies that are aimed at actually bringing back vision or more regenerative therapies. So when we think about things that are aimed at slowing down vision loss, that can fall into different categories. So kind of the most broad category that we talk about would be neuroprotection. And the idea there would be, are there medicines or nutrients that we can give that will slow down the process and prevent cells from dying? And this is something not new. We've been trying to do this for decades, but unfortunately, it's not been a very successful endeavor thus far. But I think there is, some hope on the horizon, because we're starting to understand the underlying mechanisms of why cells die a little bit better. So, just to give kind of one example of something that is in the pipeline, and actually being studied for Usher syndrome, is a supplement called N-acetylcysteine, or NAC. And this is a kind of a special kind of antioxidant, and it's actually a drug that's been approved for many years and used for different purposes. So this is a drug that we actually give people if they take a Tylenol overdose. And it turns out that if you take a high dose of Tylenol, it does a lot of oxidative damage, and you can give this drug to actually counteract that. But we've also found that if you look at animal models of retinitis pigmentosa, there does tend to be some oxidative damage. And if you give mice who have retinal degeneration NAC, it does seem to slow down the progression of the disease. So that's kind of led us to want to do a clinical trial to try this drug in people. And so in the United States and other places, there is trials that are enrolling patients in the near future, looking at giving this drug primarily to patients with retinitis pigmentosa, but that does not exclude patients with Usher syndrome. And indeed, many patients with retinitis pigmentosa have mutations in genes that also cause Usher syndrome, but they don't necessarily have the hearing loss. So that's something that'll be enrolling over the next few years, and it's going to be a little bit of time before we know whether or not it works. There's another version of this. There's another drug that is a derivative called NACA, or NACA, and that is N-acetylcysteine amide. And it's essentially a stronger version of the drug, and that's being developed by a company called Nacuity. And they're actually doing a trial already in Australia in patients with Usher syndrome. And so it's about 1 or 2 years into the trial. It's been enrolled, and we're waiting for the results. So that's something that'll be interesting to see if it shows any efficacy, because then there are even stronger versions of the drug that we can develop, and even maybe something like a small implant that dissolves the drug, so you wouldn't have to necessarily take it every day. So that's kind of one thing that's being developed, and that's actually pretty exciting. Other things in terms of slowing down the disease, I think you're all familiar with the ProQR company, which was developing a antisense oligonucleotide, which is a small piece of DNA that is designed to essentially block the mutation. And the unfortunate news was that this trial is not being continued at this point. And the reason for that really doesn't necessarily have to do with that they didn't find that it didn't work. But what happened was, the company had another trial for a different disease that failed, and they essentially ran out of money. And so they decided not to pursue further development of that. So, that's not great news, but it's also not the end. Many times what will happen is programs will get picked up by other companies. So I think that's something that could still potentially, move forward. And indeed, there was some positive data from the ProQR drug for Usher syndrome. When they studied that drug in patients, what they found was that the patients who got the drug did show a slowing of the decline as compared to patients who didn't get the drug. And it was still kind of an early result, but it was encouraging enough that I think that it's possible that another company might come and bring that drug back. So we'll kind of have to wait and see. Now, there are other things being developed, as most of the development thus far has been for type 2 Usher syndrome, specifically USH2A, and that's because that's one of the most common forms of Usher syndrome. But there's also work being done on other forms of Usher syndrome. For example, type 1 Usher syndrome, which is a more severe form of the disease, specifically type 1 Usher syndrome that is caused by mutations in myosin 7A. And there's a new company called Atsenia, and they're working on a gene therapy approach for patients who have mutations in myosin 7A. And it's a new technology. One of the problems with gene therapy in Usher syndrome is that the genes are very large, and so they don't typically fit into the gene therapy vectors that we have. But this company has a different approach where they're able to take the gene and basically cut it in half and put one half in one vector and the other half in another vector. You inject them both and they combine inside of the cell. So that's a trial we're going to see starting probably in another year or so. And so I think that's very exciting because there's really good preclinical data, to show that this gene therapy might work. For USH2A, that gene is still too big to fit into a gene therapy vector. But there might be other ways of eliciting gene therapy. So one way might be, instead of using a virus to deliver the gene, we can use something called a lipid nanoparticle, which is a little kind of ball of fat that you can put the gene inside of. And those are new techniques that were being developed to get genes inside of cells. And in fact, if you've had the COVID vaccination, that used a lipid nanoparticle to deliver the gene for the spike protein. Also, there's a whole new field called gene editing, which is a little different than gene replacement. So up to now, what we've been doing with gene therapy is putting a new copy of the gene into the cell. And you can kind of think of it like if a gene is like a cookbook and you have a mutation in the gene, it's one of the instructions is defective, and you're basically putting a whole new cookbook into the cell. And well, if the cookbook's too big and you can't get it in, that's a problem. But rather than putting in the entire gene, what we can now do is actually go and make an edit to the mistake. So actually do sort of a cut and paste like you would with a word processor. And so that's gene editing using a technology called CRISPR-Cas. And we are starting to do that now. We have one clinical trial in a different disease, but it looks like there's some promising data there. And so that's something that could be repurposed. Eventually, we could use that technology to treat larger genes, such as the USH2A gene. And so that's a very exciting technology and something, probably still, 3 to 5 years away, but, definitely in the pipeline. So those are kind of the technologies really designed to preserve vision. But what about bringing back vision? Well, there are different ways we can do that. So, probably the most common way that you've heard of would be stem cells. And so if the cells are gone, we have to bring in new cells, new photoreceptors. And how might we do that? Well, you can get stem cells, and there are different ways to get stem cells. You can actually draw a patient's blood, and you can isolate stem cells from the blood. You can grow them in a dish. You can then give them certain chemicals to turn them into photoreceptors and then grow those photoreceptors in a dish, and then eventually put them back into the eye. And that's the ultimate hope for bringing back vision. Now, that's not an easy thing, and we're working on it, but I'd say it's still a ways away from really working. That's something that we have clinical trials going on right now, but they're very early. And, mainly we're treating patients who, pretty much have lost all vision, and it's really more of a safety, approach right now. And so I think that's something that's still 5 to 10 years out, maybe even longer. But once we get that to work, then that's really going to be a game changer, because then we can really bring back vision. Now, there's another, approach, and that would be to have your body regenerate its own photoreceptors. And this is something that we didn't think was possible in the past. But if you look at other animals, they can actually do that. So if you look at a salamander and you go in and you cut out half of their retina and you come back 6 months later, they'll regrow the retina. But that doesn't happen in humans. We don't regenerate our retina. And one of the reasons is that we always thought that there aren't stem cells in the retina. So when the retina gets damaged, you can't regrow it. But it turns out that's not actually accurate. There are stem cells in the retina, but they're essentially turned off. They are kind of in this sleeping state. And we're finding that there are actually drugs now that might be able to wake up those stem cells and regrow new retina. And that's very exciting, because then you don't have to go through the process of growing them outside the body and trying to get them back into the eye. Maybe the body will just do it itself. And so there's a company called Endogena, and they have a drug that you inject into the eye, and it's supposed to reactivate these stem cells and actually grow new retina. And so we'll see, that's a pretty tall order to get that to work. But there are patients already being treated with retinitis pigmentosa, and there's no reason why that wouldn't work in Usher syndrome as well. So it's a little bit of a long shot, but if it works, it'd be really amazing. So that's something that we'll, hear more about, and in the next 2 or 3 years, we'll have to see how that trial goes. The final, way of bringing back vision would be, rather than stem cells or putting new cells in, would be something called optogenetics. And what optogenetics is it's using gene therapy to make cells that are not normally light sensitive in the retina light sensitive. So when your photoreceptors die, the cells that they connect to are not light sensitive, and so you can't really see. But if we use gene therapy to put a light sensitive protein into those cells that remain behind, we might be able to turn them into sort of crude photoreceptors and bring back some vision. And so that's very exciting, because that could apply to people who are very advanced, who have lost all their vision. And I don't think that this technology is going to be such that we're going to bring back, the same kind of vision you had when you were 15. You might bring back some crude vision, enough that you can find a doorway, or, maybe see the edge of a sidewalk. But if you've lost all your vision, even something is better than nothing. So that is another, technology that's being actively pursued by, many different companies. So that's kind of a, 30,000-foot summary of what is going on in the field. And I think I'll kind of stop there and just let people ask questions.